Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 6 from our Brock Biology of Microorganisms textbook. This chapter deals with microbial regulatory systems. So let's go ahead and get started. Cells must regulate the types, amounts, and activities of proteins and other macromolecules they make. There are some genes within the microbe that are constantly expressed, and these are called constitutive genes. And then there are other genes that only need to be expressed when the organism needs to do a particular activity, and these are called regulated genes. So we're going to talk about the regulated genes mainly in this chapter. Now, mRNA itself is a fragile molecule, and it generally has a short half-life. So once an mRNA is made, it gets degraded relatively readily. And this prevents the production of unneeded proteins. If the mRNA is degraded, then you're not going to get protein production past when you need it. And this is important because keep in mind that proteins are expensive for the cell to produce. The cell is producing proteins. Those proteins require amino acids. They require energy to form. And so if a microorganism does not need a particular protein around, it won't express that protein. It will turn off gene expression for that protein. And there are several proteins that are only necessary during certain conditions, if certain conditions are met. And this is what we mean by regulated gene expression in microorganisms. So regulation of transcription typically requires these proteins that can bind to the DNA. And these proteins will either bind to the DNA and repress transcription, turn off gene expression, or they will bind to the DNA and promote gene expression, turn on gene expression. And this protein to nucleic acid interaction may be site-specific or non-specific. These proteins bind to the DNA. Usually it interacts in a specific manner. These DNA binding proteins, these transcriptional regulators, bind in a sequence-specific manner. All right, now look at this. Here is a typical gene. So you've got your DNA, and I want to break this down for you because it can be kind of confusing, but if we go through this, slowly and methodically, hopefully you'll get a good appreciation for this gene. This is a representation of a prokaryotic or bacterial gene in particular, bacterial gene, double-stranded DNA. And look what you have. You have different elements on this DNA, on this gene. You have what's known as the promoter. You have the RBS, called the ribosomal binding sequence. You have the structural gene. This is the part of the gene that will code for protein. And then you have the terminator. This is where gene transcription ends. So I want to break this down for you again. The promoter is a sequence of DNA. It's a region of DNA upstream of the gene itself. So upstream means that the transcription will proceed towards the gene. Upstream means upstream from the gene. Right here, this is the promoter region. And at the -10 and -35 sequences, you have these consensus sequences. Consensus sequences are recognized by the RNA polymerase. Remember the enzyme that conducts transcription, RNA polymerase recognizes the -10 and the -35 sequences because there are consensus sequences on there. That means gene sequences that are conserved in evolution, and these sequences are recognized by the promoter. By the RNA polymerase. RNA polymerase will bind to the promoter because of the -10 and -35 sequences. Now, look where it says repression. This means that proteins that regulate gene expression, it's called repressors, they will bind to anywhere in this region here. The repressors bind anywhere in this region here to turn off gene expression. Activators bind upstream of that sequence of DNA. So protein activators bind here, repressors bind here to repress transcription, RNA polymerase binds here. Okay, now why is this called the -10 and -35 region on the promoter? Well, it's called that because when RNA polymerase binds to the promoter, it does not transcribe the promoter. Did you follow that? RNA polymerase binds to the promoter, so the promoter is a conserved sequence, a conserved area where RNA polymerase binds. However, transcription does not actually begin until the +1 sequence. You see where it says +1 sequence? That is where transcription begins. Does that make sense? That is where transcription begins. So you see here on the RNA that's formed, at the 5' of the new RNA that's formed, the mRNA, the first nucleotide that forms the RNA is a copy. It's a complementary base to the +1 sequence of the promoter. And then what happens is RNA polymerase copies the gene. It copies the gene, the template strand of the gene, all the way through the RBS, all the way through the structural gene, all the way to the terminator. And once at the terminator, RNA polymerase will detach from the gene, and you have a completed mRNA. And again, this is in prokaryotes, so RNA processing is not necessary. This is the mature mRNA. And you see here you have a 5' end to the mRNA, you have a 3' end to your mRNA. And this is your mRNA. Remember, you started transcription at the +1 site, and you ended transcription at the terminator site. And now you have this long mRNA. So let's discuss what is the RBS. The RBS stands for the ribosomal binding sequence. This is a conserved consensus sequence where ribosomes bind. You see ribosomes, which do what? Remember, ribosomes are the organelle inside the cells that conduct translation. They carry out the process of translation. So the ribosome will bind to the RBS, and it will proceed with translation from 5' to 3'. And you might be wondering, well, what about all this stuff? Is this stuff not going to be translated? And that is exactly right. You see all this stuff right here? You see how it's upstream? This is RNA sequence that's upstream of the ribosomal binding site. And the ribosome will bind here and proceed to the right. So this is called the 5' UTR. What that stands for is the 5' untranslated region. That means that it's going to be untranslated. This stuff is just RNA. This stuff is not transcribed. This stuff is not transcribed into protein. That's why it's called the 5' untranslated region. The ribosome binds to the ribosomal binding sequence, and then it scans towards the right. Now look at this. The ribosome scans towards the right, and it's in search of the start codon. Do you guys recall from Biology 1406 that the start codon reads AUG? AUG, which codes for methionine. That's here at the start codon. And again, translation does not begin until the ribosome encounters the start codon. Only then will translation truly begin and a protein starts to be formed. And then every 3 nucleotides, every codon will be translated from RNA to protein until the stop codon. Remember the stop codons? You could have any one of 3 stop codons, UAA, UGA, UAG, and at that point you terminate translation. You have finished making a protein, finished. And at this point, the ribosome can be released from the mRNA. Remember, a release factor binds, and the release factor promotes dissociation of the ribosomal large subunit, the ribosomal small subunit, the mRNA. Everything comes apart, right? And you may be wondering, well, what about this stuff on the mRNA tail? Is this stuff not translated? That's correct. This stuff is not translated because it's after the stop codon. So this here is called the 3' UTR for 3' untranslated region. Does that make sense? So you don't translate this portion of the mRNA just like you don't translate the 5' UTR of the mRNA. That's untranslated region. You only make protein from the start codon to the stop codon. Okay, and then you have your protein made. Now you've got your protein. And the protein can also be regulated. You could turn on a protein, you could turn off a protein. Usually you could turn on a protein by adding a phosphate group to the protein. You could turn off a protein by dephosphorylating the protein. This protein could be involved in many different processes. It could be degraded, marked for degradation. So what I'm trying to say is, just like you can control gene expression by promoting gene activation or repressing gene expression, you can also control protein activity by turning on a protein, turning off a protein, degrading a protein, etc. So you could control how a protein functions. You could control it at the gene expression level. You could control it at the protein level as well. This is, if you control a protein at the protein level, that what that is called is post-translational control or post-translational modification. And if you control it at the gene level, this is gene expression regulation. Okay, so lots of terms I'm throwing at you, but hopefully that makes sense. Now, multiple outcomes after DNA binding are possible. The binding event can block transcription. This is called negative regulation by a repressor. A repressor protein binds to the repression site on the promoter to repress gene expression. Conversely, the binding event can activate transcription. This is called positive regulation by an activator. An activator protein can bind to a stretch of DNA upstream of the promoter to promote RNA polymerase binding and gene expression activity. So what is gene expression? Gene expression is transcription of a gene into mRNA followed by translation of mRNA into protein. And remember, there are some genes that need to be regulated. Gene regulation includes the mechanisms and systems that control the expression of genes. Why would you want to regulate gene expression? Well, again, remember, regulation helps conserve energy and resources. If you don't need a particular protein around all of the time, it is in the best interest of a bacterial cell to shut off expression of that gene so that you don't have that protein around. You didn't waste the amino acids or the ATP, the energy, to make that protein in the first place. And you could conserve that energy and conserve those amino acids, conserve those resources for later when you do need those proteins around. And remember what I said, you could control the activity of proteins or enzymes either post-translationally, which means after you've already made the protein, or you could regulate it at the gene transcription level by repressing the RNA polymerase with a repressor or promoting RNA polymerase with an activator. So let's begin our discussion of gene regulation with negative control. When you hear the term negative control, you should think of a repressor, repression with a repressor. A repressor protein would bind to a repression sequence at the promoter, preventing RNA polymerase from doing its job. And usually you see this type of control for biosynthetic anabolic enzymes. So this means that, you know, it's the something that is being built, right? So you can see here for example, let me show you. It's a widespread as control for amino acid and nucleotide precursors. So the cell, for instance, the cell needs to build amino acids, right? If there is no arginine around, the cell needs to create its own arginine. So it's going to make the precursors for arginine. Does that make sense? Or if the cell doesn't have particular nucleotides around, the cell needs to build those nucleotides so it can build DNA and RNA. You usually see negative control in systems that involve those biosynthetic pathways. So take a look here. Let me give you a concrete example. A cell, over time, a cell will grow in numbers, and the total protein numbers will increase, right? So you've got cell numbers increasing, total protein increasing. And notice that the blue line represents arginine gene expression. So you've got gene expression of proteins that synthesize arginine and the precursors of arginine. Does that make sense? If there's no arginine in the environment, the cell, as the population grows, the cell's producing the enzymes required to make arginine. However, if the experimenter were to spike in arginine at this point, like actually add arginine, or if the microorganism were to come across an arginine source, arginine is an important amino acid, well then you see the negative control would kick in. The cell would stop making the enzymes that make arginine, and you see here that the arginine biosynthesis enzymes would stall out. So you've turned off gene expression of those arginine synthesizing enzymes. Does that make sense? So again, when do you see negative control? You see negative control usually when you're dealing with genes that control for amino acid and nucleotide precursors, and usually the final product of this biosynthetic pathway can repress the enzymes. So what does that mean? The product of this pathway is arginine, but too much arginine can actually shut off, can help to repress its own enzymes, the very enzymes that lead to its creation. Now, what is induction? You need to know this term. Induction is the production of an enzyme in response to the presence of substrate. I'll give you an example here. So here, you can think of it this way. An inducer is a molecule that helps turn on gene expression. You can write that down in your notes. Inducers are molecules that help a gene turn on, right? A gene to be activated. Now look at this example. By the way, typically affects catabolic enzymes. Okay, catabolic enzymes means enzymes that break stuff down. So take a look here. Again, we have our example where total number of cells is increasing, total protein is increasing, and what you see here is you have no expression of the genes for the enzymes that break down lactose. You know why? There's no lactose in the environment. There's no lactose in the environment. So what you see here is no production of lactose catabolizing enzymes. These enzymes that break down lactose are not being synthesized, right? However, if the experimenter were to spike in lactose, now there is lactose to catabolize, and the lactose serves as an inductor an inducer to an inducer to turn on gene expression, to help turn on gene expression. In this case, it's by deactivating the repressor, which we'll get into in a minute. But you add the lactose. The lactose is the inducer, and you have gene expression of enzymes like beta-galactosidase. This is the enzyme that breaks down lactose. Does that make sense? You would not need beta-galactosidase before you have lactose present. But once you have lactose present, lactose serves as the inducer, turning on beta-galactosidase expression, and beta-galactosidase is the enzyme that breaks down lactose. So let me show you this same system shown in figure 6.5. Let me show you this in a little more detail. This here is an example of what's called a negative inducible control, gene expression control mechanism. Negative meaning what? Remember from your notes earlier, I told you that negative means it involves a repressor. And inducible meaning what? Remember earlier I told you inducible means you add a small molecule that turns on gene expression. So when you see negative, I want you to think repressor. When you see inducible, I want you to think can turn on. You can do something to turn on gene expression by adding a molecule. So here's a system where the repressor binds downstream of the promoter to the Lac operator, and that prevents RNA polymerase from transcribing this Operon. And what is this Operon? This is called the Lac Operon. Lac for lactose. Lac-Z, Lac-Y, and Lac-A are genes required for proper breakdown of lactose. It's part of the metabolic pathway of lactose catabolism, right? So when there is no lactose around, the Lac repressor, this repressor binds to the Lac operator, preventing RNA polymerase from transcribing this Operon, including the 3 Lac genes. Then, when you add lactose, some of that lactose gets converted to allolactose. Allolactose is a small molecule that binds to the repressor and inactivates the repressor. The repressor then removes itself from the Lac operator, and RNA polymerase is free to transcribe the Operon, and including the 3 genes, Lac-Z, Lac-Y, and Lac-A. So do you see how the Lac Operon works? And for those of you going on to medical school, you may want to learn the Lac Operon in good detail, because I've heard it's part of the MCAT exam. So you may want to understand how this works. I may not have mentioned this earlier, but this is a good time to bring it up. I want you to know what an Operon is. Do you remember an Operon? An Operon is multiple genes under the control of a single promoter. So you see, you have the Lac promoter, then you have multiple genes, and usually these are metabolically related genes along the same metabolic pathway. So you see, you'd have 3 different genes all involved in the same metabolic pathway. In this case, the metabolic pathway would be to break down lactose. So it's very interesting stuff, because you usually don't see this in eukaryotes. Eukaryotes typically do not have Operon, which means in a eukaryote, 1 promoter is used to express 1 gene, right? 1 promoter per gene. In prokaryotes, very commonly, you'll see Operon, which means 1 promoter, several metabolically related genes along the same metabolic pathway. And yes, Lac-Z is the gene that will give you one protein. Lac-Y is a different gene for a different protein, and Lac-A is a different gene for a different protein. And when this Operon is transcribed, you're going to end up with one really, really long mRNA. And a ribosome can bind here on the mRNA to transcribe translate Lac-Z. A ribosome can bind here and translate LAC-Y, and a ribosome can bind here, translating LAC-A. Isn't that really interesting? So you have one super long mRNA, and that mRNA is used to translate multiple proteins simultaneously. It's a very quick and efficient way of getting all the enzymes you need to do a metabolic process. So in this case, if there's lactose around, you can very quickly transcribe and translate all the different enzymes you'll need to break down that lactose. So here you can see this is again what we were talking about. This is called the negative inducible gene regulation mechanism. Negative inducible. What does that mean? That means that some regulatory gene gave you a repressor, a regulatory gene expression led to expression of an active repressor. This repressor is constitutively active, which means this repressor can repress gene expression. This would be, for example, remember the LAC-Z, the LAC-Operon, LAC-Z, LAC-Y, LAC-A. And this repressor is always bound to the operator, always bound downstream of the promoter, preventing RNA polymerase from binding and preventing it from expressing this operon or this gene. So transcription is normally off. I want you to know. Remember, what does negative mean? What did I ask you to think about? Negative means it involves a repressor. Here's your repressor. And inducible, what did inducible means? It means you could do something to turn on gene expression. You add something to turn on gene expression. Remember, that's the inducer. So now what do? We add the inducer substrate. This is the small molecule that can induce gene expression. What it does is it binds to the repressor, making the repressor inactive, so the repressor can no longer repress gene expression. The RNA polymerase is free to bind to the promoter and promote gene expression. And you have your genes on. Okay, so again, very straightforward. Negative inducible. Negative meaning a repressor is involved. Inducible meaning a molecule must be added to allow gene expression. So the only way to do that, allow gene expression when there's a repressor involved, is to inactivate the repressor. I hope that makes sense. Well, let's look at some other examples. So we've looked at negative inducible. Let's look at negative repressible. That's another type, right? So what would you think if negative inducible is a system where normally gene expression is off, but you can turn it on? What do you think negative repressible means? All right, well, that negative means you have a repressor involved, and repressible would mean you could turn off gene expression by adding something. I know it's a little bit tricky, but let's try to touch on that. Okay, is this negative repressible? Take a look here. Negative repressible system. Again, negative meaning what? A repressor is involved. Repressible meaning what? This means that you can add a molecule to turn off gene expression. That means that the gene is normally expressed. All right, let's take a look here. So in this type of system, RNA polymerase can normally bind to the promoter, promoting gene expression of your transcriptional products, proteins, right? And then usually the very product of this pathway can feed back to turn off its very expression, right? So the product of the pathway itself can inhibit the pathway itself. So let's see how that would work. Again, negative meaning a repressor is involved. Repressible meaning a molecule can turn off gene expression. So again, in this case, a regulatory gene would give rise to an inactive repressor. This inactive repressor cannot repress transcription, cannot repress gene expression of this operon or gene. That means RNA polymerase is free to bind to the promoter and promote gene expression. However, This small molecule, and again, the small molecule usually being the product of this expression, this small molecule can feed back. It can bind to the repressor and actually makes the repressor active. Now the repressor can bind to the operator, preventing RNA polymerase from binding. Did you see that? So this is an example of what's called feedback inhibition. If you've ever heard of that term before, from Biology 1406, feedback inhibition. Let's look at an example here. Let's look at an example. Arginine operon. So this is called the arginine operon. Remember the arginine operon? We talked about it before. I just want to show you this. Remember arginine, as the cells increased in number, the total protein increased in number. Your arginine, remember this is normally on, this gene is normally on. The enzymes that synthesize arginine are normally on. But if you have too much arginine around, if I were to add a ton of arginine, if the experimenter were to spike in arginine, that would repress the expression of the enzymes that synthesize arginine, right? Turn off the arginine's biosynthesis enzymes. So arginine serves as the repressor. Specifically, real quick, let me tell you, it's called a co-repressor. If an inducer turns on a gene, it's a small molecule that turns on gene expression. A co-repressor is a substance that represses. It helps the repressor to repress, right? So let's take a look here. Here's your arginine operon. Here's your arginine operon. Again, the repressor is inactive. Remember, the repressor is inactive normally. So the repressor is around, but it's inactive. Again, this is a negative repressible control. Negative meaning a repressor is involved. But remember, the repressor is not repressing because it's a repressible system. That means it's a system that you can turn off by adding something. So the repressor is around, but the repressor is not doing anything. This means RNA polymerase is free to bind to the arginine promoter and to promote gene expression of genes ARG-C, ARG-B, and ARG-H. So you're forming these enzymes. These enzymes are required for the biosynthesis of arginine enzymes, biosynthesis enzymes, and so you can make arginine. Now, if I were to spike in arginine, if I were to add arginine to the system, arginine is a co-repressor. Arginine itself can bind to this repressor, activating the repressor. The repressor binds to the arginine operator, or the ARG operator, and now it gets in the way of and blocks RNA polymerase. RNA polymerase can no longer synthesize, can no longer express this gene, and so no more production of ARG-C, ARG-B, ARG-H. And it makes sense. You wouldn't want to synthesize your own arginine if there's plenty of arginine around. Again, this is an example of a negative repressible control. Negative meaning it involves a repressor. Repressible meaning it's normally on, but by adding a molecule, usually a product of that very pathway, arginine in this case, you can turn off control. Isn't that cool? Again, what did I tell you an operon is? Normally found in prokaryotes, not eukaryotes. It's a cluster of consecutive genes like LAC-Y, LAC-Z, LAC-A, whose expression is under the control of a single operator, a single promoter, single operator. All genes are going to be transcribed as a single mRNA, and transcription can be physically blocked by a repressor, and transcription can be activated by an activator, right? You've got activators for positive control, repressors for negative control. We've talked in detail about negative control, right? We've talked in detail about repressors, negative control. So next we've got to talk about activators with positive control. But first, let's take a quick little break time with Gizmo and Wicket, and we'll come back strong to talk about positive control, and then we'll finish this chapter strong by giving you some more examples of gene regulation in the microbial world. What do you say? Alright everyone, welcome back from break time with Gizmo and Wicket. Let's continue on with this chapter, chapter 6. Now recall we left off, we finished talking about negative control, which involves repressors, and we're about to talk about positive control, which involves activators. Regulatory protein activates the binding of RNA polymerase to DNA. An example is the Maltose catabolism pathway, or activation. So here is an example. You've got the Maltose promoter and the Maltose Operon. So the Mal promoter, and then here are the Mal Operon genes, Mal E, Mal F, Mal G. Now, the Maltose activator protein binds to the activator binding site, and that helps recruit RNA polymerase to the promoter. But the Maltose activator in this case is not active unless the inducer maltose is around. So this is an example of what? It's an example of a positive inducible operon. Positive meaning it involves an activator protein. Inducible meaning an inducer molecule is required for activation. And this makes sense. Let's take a step back here. What is this operon? This operon is the operon for maltose breakdown. Remember, maltose is a disaccharide of glucose and glucose. So if you have two glucose molecules and you link them together with a glycosidic bond, that's the disaccharide maltose. And there are enzymes required in the metabolic catabolism of maltose, the breakdown of maltose for energy. If you're an E. coli or another microorganism, you don't need the enzymes to break down maltose unless maltose is actually present. Because would you want to waste the resources that you need, the amino acids and the ATP, to make maltose break enzymes that break down maltose if there's no maltose around? Or would you only want to activate the expression of those genes when maltose is around, and that's when you would need them around? So in this case, the maltose activator protein is around, but it is not active unless maltose is present in the surrounding solution. When maltose is present, maltose serves as an inducer. Maltose binds to the maltose activator protein. The maltose activator protein binds to the activator binding site, recruiting RNA polymerase to promote gene expression of Mal E, Mal F, and Mal G. Does that make sense? So in this case, you only get expression of those maltose breakdown enzymes when maltose is around. So again, this is an example of a positive inducible pathway or gene expression regulation. Some regulatory gene gives rise to an inactive activator. An inactive activator, which means that transcription is normally off, but this is an inducible system. So this substrate, maltose in this case, maltose binds to the activator. The activator is now active. The activator binds to the activator binding sequence upstream of the promoter, helping to recruit RNA polymerase to promote gene expression. Now let's before I move on, by the way, activator binding sites are upstream of promoters. Remember, activator binding sites are upstream of promoters, but they could also be well upstream of promoters. They could be, well down the DNA. As long as the DNA could loop around, the activator binding site might be either right upstream of the promoter or well upstream of the promoter, because remember, DNA can loop around, and because of this, activator binding sites may not necessarily be directly next to promoters. They could be well upstream as well. Here's an example of what's called, this is the last of the gene expression regulatory mechanisms we're going to be talking about. This is an example of a positive repressible system. Positive meaning an activator is involved. Repressible meaning gene expression is normally on. So in this case, a small molecule will turn off gene expression. So you see what happens? A regulatory gene is expressed into an active repressor, an active activator. The active activator binds to the activator binding site, helping to promote RNA polymerase, and gene expression is normally on. Next, what can happen is this product can actually feed back, binding to the activator, inactivating the activator such that it doesn't bind to the activator binding site, it doesn't recruit RNA polymerase, and transcription is turned off. So you see, this is an example of turning off of a product. The product of the pathway itself feeds back to inhibit its own production. This is an example of feedback inhibition, where the product of a pathway can feed back and serve as the inhibitor for the very pathway that it helped to promote. Next concept here, this is the concept of a Regulon. If you have multiple Operon controlled by the same regulatory protein, this is called a Regulon, and the Maltose genes are an example of a Regulon. So if you were to look at the E. coli chromosome, this is the circularized E. coli chromosome. You have maltose genes here, you have maltose genes here, maltose genes here. So this is called a Regulon because you have scattered genes throughout the genome, whereas the lac operon is simply a lac operon. It's not a Regulon because all of the lac genes are localized to one loci on the DNA and under the control of one promoter. The lac operon is an example of just an operon, whereas the maltose or mal operon, there's multiple mal operon and another mal gene here, and that's called a Regulon. Now, how are those different than eukaryotic genes? By the way, everything we've discussed so far in Chapter 6 is in relation to prokaryotic gene expression. This is how prokaryotic genes, such as the genes in E. coli, this is how E. coli is regulated, right? But how about eukaryotic gene regulation? How is that different? Well, eukaryotic genes are not organized into operon, so you have one promoter per gene. Chromatin structure can affect gene expression in eukaryotic cells. Remember chromatin, you've got DNA, and the DNA is wrapped around this octamer of 8. You've got 8 different histone proteins, and then the DNA wraps around these histone proteins to form chromatin. Well, you could modify those histone protein tails. And when you modify histone proteins, that could cause the proteins to come together. The chromatin condenses, right? And the histone proteins come together. And that could cause sequestration of the promoters. The promoters get blocked. So basically, if I were to modify histone tails, and there's a promoter here, if I were to modify those histone tails, they could curl up, condense, and the chromatin can condense, and that could sequester promoters. So I could turn off gene expression simply by modifying histones and modifying chromatin. Isn't that interesting? Don't forget also in eukaryotes, you have the presence of a nuclear membrane separating the chromosomes from the ribosomes. So transcription and translation cannot happen at the same time. So in prokaryotes, recall that transcription and translation can happen at the same time in the same place. The DNA is nucleoid. It's floating around the cytoplasm. And the RNA does not need to be processed, right? You don't have the 5' cap, 3' poly A tail. You don't have intron splicing. So what that means is before transcription is even finished, you can start translation in a prokaryote. However, in a eukaryote, that's not the case. In a eukaryote, transcription has to be completed, and then the mRNA has to be processed from pre-mRNA to mature mRNA through a process of, remember those 3 modifications, a 5' cap, a 3' poly-A tail, and intron splicing, intron removal. And then the mature mRNA is escorted out of the nucleus into the cytoplasm, where it meets up with ribosomes, and only then is translation conducted, right? So, those are just a few differences. And another difference I do want to point out before we move on is that in prokaryotes, you have repressors and activators, and this is pretty straightforward little process. Whereas in eukaryotes, you have a host of different what are called transcription factors. And these transcription factors, there could be numerous transcription factors that help promote gene expression of numerous different genes. So it's quite a bit more complicated. Gene expression is much more complicated in eukaryotes than it is in prokaryotes. But luckily for us, we're not touching on that much at the moment. Global control systems. What are these? The global control systems regulate expression of many different genes simultaneously. The global control systems can include lactose operon, maltose regulon. Catabolite repression is an example of global control. Catabolite repression, so turning off catabolism. Controls the use of carbon sources if more than one is present. Synthesis of unrelated catabolic enzymes. Let me show you an example in a little bit. So let's say the cell has 2 different sugar sources or 2 different carbon sources. Which one is used first? The bacteria have preference over sugar sources. They will use the better energy source first, and once growth stops, after a lag period, the microorganism will consume the second source. So, and this results in what's known as diauxic growth, where you have 2 growth curves. So E. coli will consume the glucose first, for instance. If there's glucose and lactose present in the solution, glucose will grow off of the glucose first. It will deplete the glucose, entering a lag period, and then it will start to feed off of the lactose second. And notice how the growth curve of lactose is more shallow than the growth curve on glucose, because glucose is the preferred energy source, and that allows for quicker growth of E. coli. And then only once that glucose is exhausted is there a lag period, and probably switching on of the Operon, switching on of the beta-galactosidase and all the other enzymes that break down lactose, switching to a lactose growth curve. Let's talk a little bit about cyclic AMP and the cyclic AMP receptor here, because it is involved in regulation of the lac operon. So to fully understand how the lac operon works, you need to also understand cyclic AMP and the cyclic AMP receptor protein. So in catabolite repression, transcription is controlled by the cyclic AMP receptor protein, CRP, an activator protein, and it is a form of positive control. So this is a activator protein. CRP is the cyclic AMP receptor, and it is an activator. CRP binds to DNA only if it is bound to cyclic adenosine monophosphate, or CAMP. So take a look at this. This is your ATP. This is ATP, and you can circularize ATP. You can circularize it, so you cleave 2 of the phosphate groups off. This phosphate then binds to the 3' hydroxide hydroxyl group. The 3' hydroxyl group gets, through dehydration reaction, 3' hydroxyl group is removed, and you've circularized the phosphate group here. The phosphate group is now bound to the 3' carbon, and this is called CAMP, or cyclic AMP. Cyclic AMP is the effector molecule for the CRP protein. The CAMP, is it exactly called? The CAMP receptor protein. So cyclic AMP, the level gets high enough, it binds to the CAMP receptor protein, then the CRP binds to the lactose operon. Let me show you how that works in the next slide here. So you've got your CRP protein. When it binds to CAMP, cyclic AMP, it can dimerize and activate. Now you have your activator, activated activator, binding to the LAC operator at the CAMP or CRP binding site. This promotes binding of RNA polymerase to the promoter of the LAC operon. And you have gene expression. You can have gene expression of LAC-Z, LAC-Y, LAC-A. Remember the LAC operon. Remember this repressor for the LAC system. If the LAC repressor is activated, the LAC repressor can repress activation of the LAC operon, and if allolactose, the inducer, is around, that makes the LAC-I, the repressor, inactive, allowing for gene expression of the LAC operon. I know it's a convoluted system, but recall that you may want to focus on this, maybe read up on this from your textbook. If you're going to medical school, if you're gonna study for the MCATs, they expect you to know the LAC operon in detail. Like, how do you turn on the LAC operon? Is it an example of a negative inducible or a negative repressible system? What is the role of CRP protein and CAMP in this operon, etc.? All right, now let's move on from that basic kind of control and talk a little bit about post-translational control, like how are proteins post-translationally controlled, and also how does signal transduction work inside of prokaryotic cells. Let's talk about this. So prokaryotes regulate cellular metabolism in response to environmental fluctuations. The signal may be transmitted directly to the target, or it may be detected by a sensor, like a receptor, and transmitted to a regulatory machinery. This is called signal transduction. So I'm going to introduce this concept of signal transduction. Most signal transduction systems are two-component regulatory systems. The two-component regulatory systems include usually a sensor kinase and a response regulator. A sensor kinase, by the way, before I move on, a kinase is an enzyme that can add a phosphate group to things. So a kinase is an enzyme that adds a phosphate group. And in the world of molecular biology, in the world of biochemistry, oftentimes by adding a phosphate functional group to a protein, you have then activated the protein. And by dephosphorylating the protein, taking the phosphate group off of that protein, you have deactivated that protein, usually. So let's see how this works. So a sensor kinase is like a receptor, a cytoplasmic membrane receptor. The receptor will detect something in the environment, and then it can auto-phosphorylate. It can phosphorylate itself once it detects this environmental signal. And then a response regulator in the cytoplasm can respond to that phosphorylated kinase, and as a result, it can bind to DNA and regulate protein transcription. So let's take a look here. You have the plasma membrane, so the membrane of the cell. In the plasma membrane, you have these sensor kinases. Remember these sensor kinase proteins, the transmembrane proteins? They can bind to some kind of environmental signal, something that the bacteria wants to respond to, and then ligand binding to the receptor, This causes autophosphorylation on a histidine residue. You see, you have histidine. It gets phosphorylated. ATP comes in and phosphorylates. It gives the terminal phosphate to the receptor. Now the receptor is phosphorylated. That means that the receptor is activated. Okay? And this is recognized by a response regulator. Did you see that? The cytoplasmic protein response regulator recognizes the phosphorylated sensor kinase. That causes phosphorylation, so the phosphate group is transferred to the response regulator, activating the response regulator. The phosphorylated response regulator shuttles to the gene operator, binds to the DNA operator, and in this case it would block transcription, because it's binding to the downstream of the RNA polymerase. Transcription is blocked. So this environmental signal resulted in turning off gene expression of this gene in this way. So you see how that's a two-step process, a two-component regulatory system. Component 1 is the sensor kinase. Component 2 was the response regulator. The environmental signal bound to the sensor kinase. Sensor kinase was autophosphorylated, which led to phosphorylation of a response regulator. Response regulator served as a repressible, negative repressible system, right? Because you've got the response regulator is a repressor, but it only can repress once activated by a phosphate group. Okay, very interesting stuff. Let's keep going. So in E. coli, you have almost 50 different two-component systems, like the one I just mentioned. And these include processes of phosphate assimilation, nitrogen metabolism, osmotic pressure response. All of these are systems where two-component systems play a role. So how do cells, for instance, regulate chemotaxis? You remember earlier we talked about chemotaxis and other forms of taxis. This is directed cell motility, where a cell can move towards an attractant or away from a repellent. And this has to do with counterclockwise rotation of the flagella for run and clockwise rotation of the flagella for tumble. And remember how motility works for microorganisms. It works as a series of runs and tumbles, runs and tumbles. The bacteria will run towards the attractant and tumble if it's moving away from the attractant, eventually making its way towards the attractant. So how does this process work? How does chemotaxis work? Well, in response to a signal, a sensory protein in the cytoplasmic membrane sense the attractants and repellents and interact with cytoplasmic sensor kinases. The cytoplasmic membrane protein, the membrane receptor, you can think of it, is called Methyl-Accepting Chemotaxis protein, or MCPs. MCPs are the membrane receptors, the membrane proteins that respond to the attractant or the repellent in the environment. So binds attractant or repellent to initiate flagellar rotation. Remember, counterclockwise runs, clockwise tumbles. And the way it does it is by interacting with CheY proteins. CheA sensor kinase, that's the kinase, and CheW. And to control the flagellar rotation, CheY protein plays a major role. CheY results in runs, whereas phosphorylated CheY results in tumbles. So let's take a look here. I'm going to show you this image. This image involves your cytoplasmic receptor protein, right? This is the methylated protein I told you about, methyl-accepting protein. This is the receptor kinase. Then you have your regulatory kinase, CheW and CheA. CheA is the one that gets phosphorylated. And downstream of that, remember, is your CheY, which can become phosphorylated as well. So let me guide you through this. I'm just going to show you about the flagellar rotation, this pathway here. So if there's decreased attractant binding to MCP, decreased, so this is attractant, these are attractant molecules, like let's say glucose, for instance, attractant. Decreased attractant binding to MCP triggers phosphorylation of CheA, CheW complex. So this results in CheW and CheA getting phosphorylated. CheA, CheW phosphorylation results in phosphorylation of CheY. So MCP doesn't bind as much attractant. CheA gets phosphorylated. This results in CheY getting phosphorylated. So now you've got phosphorylated CheY. CheY can bind to the, the motor in the basal body of the flagella. CheY, phosphorylated CheY binds to flagellar switch, and that switches the flagella to clockwise rotation. Tumble time. CheZ can then dephosphorylate CheY after a moment. CheZ dephosphorylates CheY back to CheY. CheY, when it's unphosphorylated or dephosphorylated, can no longer bind to the flagellar motor proteins, and we switch back to a counterclockwise run. Do you guys follow that? When there's decreased attractant, MCP promotes phosphorylation of CheA, CheW complex, which then results in phosphorylation of CheY to phosphorylated CheY. Phosphorylated CheY can bind to flagellar motor proteins, the flagellar switch it's called, switching the flagellum to a clockwise rotation to induce tumble. After a bit of tumbling, CheZ can dephosphorylate CheY, phosphorylated CheY to CheY, regular CheY. Regular CheY is not able to bind to the switch protein, and the flagella reverts back to its preset run, counterclockwise rotation. You guys follow that? Very interesting. So this is an example of the attractant promoting runs and tumbles. How does the bacterium, think about it, if the bacterium is moving away from the attractant, you want to stop running away from the attractant. You're running away from the attractant. You're running away from what you want. You're going to want to trigger a tumble so that you can get reoriented and maybe hopefully start making your way next towards the attractant. Now you might be wondering, what is the role of CheB and CheR? This has a role in what's known as adaptation. Let's see if I can define adaptation for you. So adaptation, once an organism has successfully responded to stimulus, it must stop responding and reset the sensory system to await further signals. This is known as adaptation. So adaptation makes it so that this response stops. So you stop responding to signals. Let's say you've made your way to the attractant. You've made it into the attractant. You don't want to continue to run and tumble. You want to just enjoy this food, right? You want to enjoy the glucose. So adaptation allows you to deaden this system so that you stop running and tumbling because you've reached the system. So this involves CheR. CheR methylates MCP, and fully methylated MCP is no longer, responsive to the attractant. In fact, it becomes more responsive now to repellent. So what happens is when CheR is constantly methylating MCP, but phosphorylated CheB can demethylate MCP. Phosphorylated CheB can demethylate MCP. So when the attractant is low, remember when the attractant is low, this activates MCP to phosphorylate CheA. CheA then phosphorylates CheY, remember, but it can also phosphorylate CheB, which demethylates MCP. But if the attractant concentration is high, you don't get activation of CheA or CheW, which means you don't form CheB. CheB is dephosphorylated, so the only thing going on is methylation of MCP, and eventually you deaden MCP because it becomes fully methylated. All right, so other forms of taxis involve phototaxis, movement towards the light. MCPs play a role here, and the Chi proteins play a role here. Aerotaxis, again, plays a role here as well. Now, the last concept of this chapter is quorum sensing. It's a very interesting type of signaling and control of expression that occurs in prokaryotes. And this is prokaryotes that can respond. Quorum sensing involves prokaryotes that can respond to the presence of other cells of the same species. Long ago, it was just assumed that microorganisms cannot communicate with one another. Does that make sense? So a bacterium doesn't have eyes, it doesn't have ears, it doesn't have a smeller. So it was assumed that bacteria cannot communicate with other bacterium, but that was an incorrect assumption. It's been shown that bacteria can actually communicate with one another. And the way they communicate with one another is through this process of quorum sensing. That's a mechanism by which bacteria and some archaea assess their population density. So they can tell whether there are several of their species around, or if they're alone, or if there aren't many of themselves around. And why would you want to do this? Well, this ensures that a sufficient number of cells are present before initiating a response that, in order to be effective, requires a certain cell density. So, for example, producing a toxin. Why would you put the effort into producing a toxin if there aren't enough of you around for that toxin to have an effect? Or bioluminescence. Let me show you. Bioluminescence. There are bacterium that glow, and they play a role in fish, for instance. They're part of organ systems in fish which glow, and the fish or squid can benefit from that glowing, and in order for the bacterium to put in the effort to glow, they need to be aware that there are other of their own species around to make the glowing worthwhile, right? Why glow if you're a single bacterium? You're not going to achieve anything. But if you're part of a fish and you glow, then you're gonna help the fish to feed. That's going to bring more nutrients to the fish, which in turn brings more nutrients to you as the bacteria living on the fish. Does that make sense? Anytime bacterium want to do something that benefits them only when they're in sufficient numbers and not when they're alone, those are systems that are controlled by quorum sensing. So how does quorum sensing work? Quorum sensing works because each species of bacterium that's able to do quorum sensing, not all bacterium can do quorum sensing, by the way. Each species of bacterium that can undergo quorum sensing produces a specific autoinducer signaling molecule. This is a molecule that each bacteria can release into the environment, and other bacterium in the environment can sense that molecule. So this molecule diffuses freely across the cell envelope into the environment. Once this molecule reaches a high enough concentration inside the cell, again, this is only if many other cells are nearby making the same autoinducer, this can trigger binding to specific activator proteins or sensor kinases, triggering transcription of specific genes. Isn't that cool? So take a look here. Here's a bacterium which is producing the autoinducer. It's called AHL, Acyl Homoserine Lactone, AHL. The bacterium is producing the autoinducer AHL. But this AHL in the environment is not sufficient concentration to do anything. However, if there are other bacterium in the neighborhood of the same species, this bacterium is also producing the same AHL, right? Acyl, Homoserine, Lactone. Here's an example of what this organic molecule looks like. It's a small molecule. This bacterium is releasing AHL. This bacterium is releasing AHL. This bacterium is releasing AHL. They're all releasing AHL. At some point, you're going to have sufficient concentration of AHL, of this bacterial species-specific AHL, to bind to the activator protein. And now this activator protein can bind to specific promoters on the bacterial chromosome, promoting gene expression of quorum specific proteins. And now you have these quorum specific proteins to do the quorum specific task, which is, again, like make a toxin or be involved in bioluminescence. Isn't that cool? So several different classes of these autoinducers are created. Remember these acyl, homoserine, lactones, AHLs? They're also called autoinducers. The other species make autoinducers. There's autoinducer 2, autoinducer 3. Again, every different species makes its own autoinducer. There is AHL, there is AI2, AI3. Gram positives can make short peptides as autoinducers. Autoinducers, there's a myriad different autoinducers. But what do they have in common? Your species makes a particular autoinducer, releases it to the environment. If there's sufficient population of you making this autoinducer, the autoinducer will reach a specific threshold concentration in order to bind to a specific receptor kinase or regulatory molecule, activating that regulator to turn on gene expression. So quorum sensing, first discovered as mechanism regulating light production in the bacteria, including Vibrio fischeri, which is really cool. Vibrio fischeri is a bacterial species that can bioluminesce. And it bioluminesces once it activates what's called its Lux Operon. This Lux Operon contains genes, which are enzymes that encode for bioluminescence. So take a look here. Here's a squid. And this squid has, on its underside, basically they call it the third eye, but it's a region where these bacterium, Vibrio fischeri, can grow. And it's almost like an incubator. The squid serves as almost like an incubator for these Vibrio fischeri bacterium. And these Vibrio fischeri grow in this little pocket in the squid. And because they're so concentrated in population, their autoinducer reaches a certain level, triggering the Lux Operon. The Lux Operon produces bioluminescence, and when the squid swims around at night, it can use this bioluminescence as kind of like a flashlight, and this flashlight dazzles creatures on the bottom of the ocean, and it hides its shadow. So these creatures that would otherwise hide, when they see a shadow, they don't hide, and they get eaten up by the squid. Isn't that an awesome type of mutualistic symbiosis? And the more food the squid gets, the more these Vibrio fischeri get food. Very interesting, isn't it? Here's another example with Escherichia coli. Now I want you to know that E. coli is a very harmless bacterium. It's a gut bacterium that everyone has in their guts. However, there is a dangerous strain that you need to know about. It's called the O157H7 strain of E. coli. This is a strain that's dangerous because it can produce Shiga toxin. It has the gene from Shigella, which codes for this toxin. So when this E. coli is growing in your gut, it's actually producing a very dangerous toxin from another bacterium, Shigella, and the Shiga toxin can give you dysentery, bloody diarrhea. It can lead to death. It can actually pretty easily lead to death if not treated correctly. So how does this E. coli quorum sensing work? This E. coli is very interesting. It produces an AHL, Autoinducer 3, AI3, which induces virulence genes, but only if gut hormones, epinephrine and norepinephrine, are around as well. Isn't that interesting? Take a look here. This E. coli will produce toxin, but only if sufficient quantities of the autoinducer from other E. coli is present, and intestinal hormones are present as well. Remember, epinephrine and norepinephrine. If both autoinducer 3 from other bacterium and intestinal hormones, epinephrine and norepinephrine from the gut bacterium, is around, that activates sensor kinases, which activate transcriptional regulators, which activate gene expression of the toxin. And now you have the enterotoxin causing damage in the gut of the animal, and can lead often to death, to animal death. Okay, so these quorum sensing, quorum sensing is a potential drug target, because if you think about it, if you come down with O157H7 strain of E. coli, couldn't you take a drug, maybe take a drug that prevents activation of these sensor kinases, these transcriptional regulators that lead to the toxin? Better understanding of how quorum sensing works could lead to drug design to disrupt this process and potentially prevent the virulence gene expression in the first place for these types of conditions in the future. All right, lastly, let's talk about biofilm formation, which is a type of quorum sensing. Remember, biofilm is when a bacterium, I should say a population of bacterium, start to produce a filmy substance. It's called the biofilm. It's a sugary secretion. Bacteria settle on a surface. There's sufficient population. They start secreting a sugary substance into the environment. That sugary substance becomes their new environment where other bacterium can attach. They all produce sugary substance, and that becomes a biofilm. Think about the plaque on your teeth. That's a classic example of biofilm. This is a gooey substance. But think about it. Bacterium would not want to make the biofilm if they're all by themselves. They want to be in groups in order to form the biofilm. So an example is Pseudomonas aeruginosa produces a polysaccharide, this sugary substance that increase pathogenicity and antibiotic resistance. So pathogenicity, because the bacterium cannot be found or phagocytosed inside the body. Antibiotic resistance, because antibiotics can't really penetrate the biofilm very well. So there are two quorum sensing systems here. You could produce AHLs and cyclic di-guanosine monophosphate, c-di-GMP. This leads to exopolysaccharide production and flagellar synthesis. So take a look here. You've got Pseudomonas aeruginosa binding to the surface, not yet in sufficient population, but now you have more bacterium attaching in sufficient population to produce the AHLs and the c-di-GMP. This promotes what? Secretion of the polysaccharide and flagellar formation. And then the bacteria the biofilm matures, and now you have your biofilm, your plaque, for instance, your biofilm. And that leads to the end of this chapter. Very interesting chapter, a little bit long, but hopefully very educational, very interesting stuff. As always, please leave any comments you have in the comment box below, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.